Welcome. Today we'll be talking about analysis of binary loading into a surfactant bilayer using Gibbs ensemble Monte Carlo simulations and ideal adsorbed solution theory. This work was conducted when I was a postdoc at the University of Minnesota with J. Elias Seaman, and now I'm currently an assistant professor at Northeastern University. Why are we interested in this topic? There are multiple reasons why understanding the uptake of solutes into a surfactant bilayer could be useful. One, we can understand and look at, for example, hydrophobic ligands being loaded into a micellar structure, and this can help with drug delivery. But we can also look at perfume raw materials and see how detergency is being affected, and also drug delivery, and understanding how we could best distribute ligands within the human body. Now, some of the questions that we're interested in studying today is, first, how do different solutes affect the structure of micelles and the Langmuir monolayers? How do they distribute in these micelle and Langmuir structures? And finally, how do the trends differ based on different solutes? Are we going to see a different distribution and different types of interactions? So today we're going to be talking about two different kind of systems. First, we're going to be looking at what occurs when we load just one solute type at a time into a surfactant bilayer. And then eventually we're going to look at what happens when we load two different solute types into a surfactant bilayer. Let's begin. So how did we set up the simulations to at least look at one solute uptake? We did this using Gibbs ensemble Monte Carlo simulations. Okay, so let's begin. We had a system. We had two thermodynamically connected boxes. One box had the surfactant bilayer with the water bead, and the other box had the vapor of the solute of interest. So this is kind of like emulating the test tube that you guys can see. So imagine our mesophase being in the bottom of the test tube, and then our vapor phase in the top. Now, in this simulation, in the surfactant bilayer box, we had 538 water beads, 100 surfactants. And in the vapor, based on the solute of interest, we ranged anywhere from 50 to 800 solute molecules, you can say, in the vapor box, and we only allowed the solute molecules to swap back and forth. Now, for this simulation, we used a force field known as STK, and this is a coarse grain force field, and you can see the different beads that we use, at least for the surfactant. And on top of it, for this Monte Carlo simulation, because it's a Gibbs ensemble, again, two thermodynamically connected boxes, we allowed the boxes to fluctuate in volume. We only allowed the solutes to swap back and forth, and we changed the vapor pressure of the solute of interest to control chemical potential. So with that in mind, we were able to obtain a few bits of information, as you can say. First, you guys can see that there are plots on the left and actual snapshots of the system on the right. So on the left, we have the first plot, which is the one of nonane loading. We looked at the partial pressures of nonane ranging from 4 to 64, or actually 2 to 64. And we looked at obtaining Henry's law, and we were able to fit to the lower partial pressures. And this is what we observed. So for nonane, as we increase in the partial pressure of the system, we saw a positive deviation. Now, looking down at hexanol, that is also, again, what we observed. And just something else to keep in mind, we see that hexanol loads on average 3 times more than nonane. And now to give you guys kind of a visual of the actual system, we see snapshots here at 4 and 16 and 64% of partial pressure. And you guys can observe that at 64%, there's a lot more of the solutes present. So more solutes load as we increase the vapor pressure. Now, we also wanted to know how these solutes were, what was the distribution within the bilayer. And we were able to do that by plotting also the density Z profile. That means, imagine here, as you guys can see, the bilayer kind of in the snapshot. So imagine that we plot the density profile so that we're getting a cross section of that bilayer. So two observations to be made. Number one, we see that nonane loads mostly in the center of the bilayer, so between the bilayer leaflets. This makes sense because nonane is mostly hydrophobic and preferred to stay within the hydrophobic region of the surfactant bilayer. Now, the hydrophilic region, which is the region near to the water, is where we observe some of the hexanols loading. So you get the hexanols load in between the individual surfactants. So again, you have your individual surfactants, kind of like my fingers here on my hand, the hexanols load in between, and the nonanes load on top. So the question now lies, what occurs when we have both solutes being loaded into our surfactant bilayer? So just to show you guys here, you can see with the image, again, nonane is loading on top, hexanol is loading in between the individual surfactants. Is there a kind of synergy, an enhancement in loading that occurs if we allow both solutes to load? So this leads to our next question. What occurs with binary loading? So we set the system up in the simulations in a similar fashion as we did with the unary. So instead of having a single test tube now, we have a kind of a test tube in the shape of a U. Our mesophase is on the bottom. On one side of the U, we have one solute of interest. On the other side, we have the other one. So in this case, we have hexanol on one side, nonane on the other. Again, surfactant bilayer of 538, like of 100 surfactants. In the mesophase, we had 538 water beads, and then 2 extra thermodynamically connected boxes, so a total of 3, and we only allowed the solutes to swap into the surfactant bilayer. And here you guys can see on the bottom snapshots of some of the systems that we obtained. We took basically the partial pressures of hexanol from 2 to 32 and nonane from 4 to 64%, and we did all the different combinations. Just a note to keep in mind, you guys can see that at 64% nonane, 32% hexanol, we have a lot of loading of the solutes. It almost appears like the bilayer is starting to come apart. Now, so again, we did this, and the first thing that we did is we obtained the number of solutes loaded at each partial pressure in all the different combinations. So we have on the left-hand side, we have the nonane system. We have the unary nonane that you guys already observed. We have our Henry's law. We also have the unary nonane basically fitted. And on top of that, we have all the different number of nonanes in the presence of the different partial pressures of hexanol, ranging from 2 to 32%. And the first thing you guys can observe is the fact that with the presence of hexanol, there is more nonane. And then you can observe the same thing for the hexanol plot on the right. Okay, so again, we have the unary hexanol, we have the line fitted for the unary hexanol, and then all the other data points in the presence of the different nonane partial pressure percentages. So the question is now asked, do we actually have enhancement? Is there an added level of synergy that occurs in the presence of both the different types of solutes? We want to now define enhancement. What is enhancement? Well, basically we want to see if the nonane to hexanol interactions are more favorable than the nonane to nonane interactions or hexanol to hexanol interaction. So we began by doing this. We look at a specific system, 32% nonane and 8% hexanol. So we obtained first the number of solutes that were loaded in the unary system. So for nonane at 32%, it was 9 nonanes. For hexanol at 8%, it was 11 hexanols. So now the question that we pose is, what happens if we, instead of having 11 hexanols there, we had 11 nonanes? So then we go and using our quadratic fit, we obtain the partial pressure that correlates with 11 nonanes, and that occurs at 37.5% partial pressure nonane. So now we have 9 nonanes at 32, 11 nonanes at 37.5. So now we compare what occurs if we now combine those partial pressures and we look at 69.5. Using quadratic fit, we see that there are 25 nonanes, while if we just added them separately, we would see that 9 plus 11 is 20. So now we know that if they're separate, that's 20, but if they're combined, right, if they're at 69.5% and they're in the presence of each other, then it's 25 nonanes that load. But the question is still posed, is that more favorable than actually doing the binary system? So now we look at the binary system. So again, 32% nonane, which gives you 9 solutes, and 8% hexanol, which gives you 11 separately in the unary system. But in the binary, they both go up by 1. So now we can have a very distinct example of nonane going up by 1 nonane, so going from 9 to 10 in the binary. And in our pseudo-binary, we see that it goes up a total of 5, but we want to know how much of that 5 solutes is a contribution from the 9 nonanes. And so we do the math, and we see that that actually goes up by 2.25 solutes. And so by taking the difference, we see that 1 minus 2.25 is negative 1.25, which means that there is basically more of an enhancement in the presence of nonane than there would be in the presence of like just hexanol. So the pseudo-binary has a higher enhancement than the binary system. And so with that math, we are now able to go and do it for all the different simulations that we ran. And we observed that mostly for nonane, it is actually, there isn't an enhancement in the binary. And for hexanol, there are some positive numbers, but barely. And so for hexanol, there isn't really much of a difference. So our conclusion is there really isn't an enhancement in the binary system. So nonane to hexanol interactions are not more favorable from nonane to nonane and are maybe equally favorable to hexanol interactions. The next thing we do is we actually use ideal adsorbed solution theory to see what is happening with our interactions. So AIChE actually looks at predicting the interactions based only and solely on the unary. So what we have here is very interesting. Looking at the nonane, we see that at lower partial pressures, it's over predicting the loading, which means based on just nonane to nonane interactions, it's expecting for there to be more of the nonanes loaded compared to what we have here with our nonane to hexanol piece. Now, on the other hand, look at the hex part of it, it predicts it pretty well. So again, we can conclude from looking at these plots that nonane to nonane interactions are definitely way more favorable than, and way more not being very quantifiable, are favorable more, are favorable than the nonane to hexanol interactions. And so we can look at that, at the AIChE, but also we were able to obtain the free energy of transfer based on our density profiles. And, the takeaway message here is for nonane in the unary system for the same number of solutes, if we compare it to the binary, it's more favorable. And for hexanol, we don't really have a discernible trend. We were also able to obtain the different locations of where nonane and hexanol loaded, and they were similar to what we observed in the unary system, nonane between the leaflets, the bilayer leaflets, and hexanol in between the surfactants near the water. And we were able to obtain other structural information. So in conclusion, with these simulations, we were able to obtain and understand to see if there is actual enhancement when both two different types of solutes were loaded into the surfactant bilayer. By initial glance, you might think there is, but we actually did the analysis and there really isn't. And with these simulations, we were able to see the structural impact we have on the bilayer. We were able to also observe and obtain the free energy of transfer. So with that, I thank you very much for listening, and I have a special acknowledgement thank you to my postdoc advisor, J. Elias Seaman at the University of Minnesota. It was a very great time, and I grew a lot scientifically there. And the group part of also all the funding sources that are here. But also, I want to bring attention to the fact that now I am a professor at Northeastern University, and I'm building up my lab, and we are known as the Combine Lab, which stands for Computational Modeling for Biointerface Engineering. I'm always looking for bright young graduate students, postdocs. If anybody is interested, please send me an email or check out our website at minkaracombinelab.com. And we have basically, because of COVID, I built most of my team virtually, and so we have our group photo here from the Zoom chat. So thank you very much for listening, and have a wonderful day.